Namaste students, I am JB Dr. Ajizman from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapeeth Women's University, Jaipur. Today I am going to discuss about the topic chromatography separation and some techniques will be discussed here. So in this lecture, the first technique which I am going to discuss about the chromatography is affinity chromatography. What is the affinity chromatography? Affinity chromatography separates the protein of interest on the basis of the reversible interaction between its antibody coupled to a chromatographic bead. So, what is actually the affinity chromatography? We know that a protein is there. Okay, everything which is present in our human system is either self molecule or a non-self molecule. Okay. If they are self, then there are some antibodies which will bind to the non-self molecules. Okay. Foreign pathogen, foreign bacteria, foreign fungus are entered inside our body, then the antibodies will bind to those particular particular bacteria, fungus, and viruses. So, on which portion the antibody bind to uh, bacteria? The antibody have a specific uh, portion on which it will bind. That is known as the epitope. And what is that epitope? The epitope is usually a carbohydrate molecule, a lipid molecule, or a protein molecule. So this protein molecule is neutralized by the antibody. So our antibody will bind to those proteins which are present on the surface of the bacteria. And this is the in vivo system. Here we can use we understood one thing, that antibody have a specific affinity to a particular kind of antigen and particular kind of protein. So we can take the use of this property of the antibody in separation of some proteins. We can use the property of this, we can use the property of the antibodies in separation of some particular proteins. Okay. So suppose that if we have some protein or enzyme which are present in our body, and we have some antibodies against those particular proteins. So we can prepare those antibodies, and these antibody will bind to particularly only to those particular proteins also. So if we have prepared these antibodies, they will bind to them, and sometimes they can be used for their separation also. So that we are going to do in the affinity chromatography, in which the protein which is specific, and to that specific protein a particular kind of antibody will be specific, and the interaction is very specific, which will lead the high interaction rate. Okay, if they are very specific to each other, and there are some 3, 4, 5, 6, 10 kinds of proteins are there, then antibody will bind to only its specific protein only. They will not bind to all those 9 proteins. It will bind to only one single type of protein. So binding to that single type of protein will lead to the separation of the particular kind of protein, which is known as the affinity of the antibody to a particular antigen. So this system is used in the separation of the particles. So with what are the some basics of the affinity chromatography? So with high selectivity and high resolution and high capability for the protein of interest, the purification level in the order of several thousands fold are achievable by the affinity chromatography, because they are highly selective. They are highly, they have high capacity for the protein of interest. They have a high binding capacity for the production of the protein. The protein of interest is a collection of the protein of interest is collected in a purified concentrated form. Okay, and the biological interaction between the antigen and the protein of interest can result from the electrostatic interaction. The binding between the antigen and antibody can be like a electrostatic interaction. Their binding may be any kind of van der Waals interactions, which are the poor bonding, and hydrogen bonding is also found between the antigen and antibody. So, to elude the protein of interest from the affinity which the interaction can be reversed by changing the pH. Okay? We can also bind the antigen antibody, and we can also dissociate the antigen and antibody. Okay, concentrating effects which enables large volumes of to be processed is is belonging to the antigen and antibody. The protein of interest can be purified from high level of contaminating substances. The antibody which are prepared to the protein of interest is very expensive. So affinity chromatography is least economical choice for the production of the chromatography. This is the disadvantage because it is expensive. It is beneficial, but it is expensive method. So this is the diagram of the affinity chromatography. In this affinity chromatography, suppose that there are three or four different kinds of proteins are there, and we have a single type of antibody which is specific to our desired protein. So our desired protein, suppose desired antibody is this red color antibody. So it will bind to only one this single type of antigen, blue color. Okay, our desired antibody which is in red, so it will bind to only this blue colored antigen only. It will not bind to any other and any other protein. So suppose this blue color is a protein and it is our desired protein. So we have some antibodies which will bind to only this desired protein only, blue color. Okay. So this blue color protein our antibody will bind and it will be eluted, it will be separated. Okay. And how they are dissociated from each other. The dissociation of antigen and antibody is conducted by change in the pH. If we change the pH, then our desired protein and desired antibody will separate it from the each other, and our product will be achieved. Next chromatography, which I am going to discuss about that, is liquid chromatography. So, what is the liquid chromatography? In liquid chromatography, the mobile phase is liquid. Okay. It is liquid in nature, so it will carry our desired protein inside a chamber for their separation. And separation is conducted at high temperature conditions in liquid chromatography and whatever the process. The liquid chromatography is a very, very scientific technique which is dependent on some specific instruments. It is carried out in some column. So the column is a stationary phase in liquid chromatography, which may have a high temperature, and the mobile phase is liquid, and the uh, column or plane is a stationary phase in the liquid in which our sample is loaded. So the sample in the liquid chromatography is added by injection, small micro injection, which may have auto sampler or which may not have any kind of auto sampler. inject our sample by injection, by micro injection. The technique which is known with the liquid chromatography is high performance liquid chromatography. High performance means it is very specific. That's why it is known as the high performance liquid chromatography. In high performance liquid chromatography, the sample is forced through a column. Sample is forced through a column that is packed with a irregular, spherically shaped particles or porous stationary phase. Okay, it is passed by it is forced through a column. Okay, column in which our stationary phase is there, and the stationary phase is a porous monolithic layer, and the pressure is conducted by the our mobile phase, that is the liquid. So in very high speed, the sample is passed by the mobile phase, that is our liquid, or the liquid which we can use is a solvent. Okay. The solvent which is very specific to our sample. For example, here we can use methanol, which is very well dissolved. It can be dissolved in our methanol, which is our solvent. So one solvent is used in which the sample is easily soluble, and this solvent will carry the sample at high speed to the stationary phase in a column, which is monolithic layer. When there is a high temperature conditions are there and they will evaporate, then their retention value will be calculated. Okay. Then according to that retention value and their mass, their molecular weight, there will be a detector which will detect which kind of molecule is this. So it will be used for the detection. It will immediately detect what is the molecule which is present in the sample. So this is a technique which is very high efficient and highly appreciable technique. So I hope the students will better understood about high performance liquid chromatography. So in this lecture, I discussed about the affinity chromatography, the basic principle, the knowledge, basic introduction, basic principles, and steam base, and what is the basic mechanism by which the molecules are separated in the affinity chromatography. Similarly, I discussed about the liquid chromatography, what is the mobile phase and what is the stationary phase in the liquid chromatography, and what is the mechanism of the separation of the particles in the liquid chromatography, that is the high performance liquid chromatography. So the question which can arise according to this lecture is that explain affinity chromatography and their principle and their mechanism of the separation. Similarly, in the liquid chromatography, a question which can arise, explain high performance liquid chromatography, their mechanism of action and their basic principle. So I hope you can answer all these questions. So my lecture is over here. Thank you very much.